This is the 31st video presentation for the Case Plant course. As you view this video, make notes of important information. Pause, repeat, and resume the video whenever necessary. The Seed, Lesson 7.3, Kernels of Life. You just completed Lesson 7.2, Pollination and Dispersion, which connected the concepts related to plant genetics to how plants physically reproduce. This presentation provides an overview of seed structure and introduced you to the germination process. Pollination and fertilization was discussed in Lesson 7.2, Pollination and Dispersion. Following fertilization, the egg develops into the embryo and the ovule becomes the seed coat. The difference between monocotyledon and dicotyledon seeds is marked. This illustration and further reading about seed anatomy is found on pages 59 and 60 in the Plant and Soil Science Fundamentals and Application textbook. A cotyledon is the seed leaf, or first leaf, that emerges from the seed during germination. Usually, cotyledons do not strongly resemble the mature leaves of the plant. Mono, meaning one, and di, meaning two, refer to the number of cotyledon. You should see that the monocotyledon seed has one cotyledon, or leaf, compared to the dicotyledon, which has two. This is critical for identification and classification of plants. The epicotyl picture is actually the next set of true leaves, which will emerge later. Endosperm is associated with monocotyledon seeds, or kernels for grasses. The starch food reserves are used by the germinating seedling until the cotyledon and the first true leaves produce enough energy for the seedling through photosynthesis. Dicotyledon seeds do not have an endosperm reserve. Instead, the starch food reserves are held in the cotyledons. Essentially, the epicotyl is a region of an embryo. The epicotyl will form all the parts above the first node of the seedling, including the first set of true leaves. These and all subsequent leaves will look like the leaves of the plant you are more familiar with. The hypocotyl will form the lower stem of the seedling, mainly the section between the and the radicle, which is the first root structure formed during germination. The seed coat, also known as the testa, provides protection. Some seed coats are very hard and require an external force to allow water to enter. Most people do not realize the seeds are living in the state of dormancy. You will be collecting evidence that seeds respire in an upcoming activity. When environmental conditions are appropriate, the process of germination will begin. Water is required for the seed to swell and break the testa. Mitosis occurs to rapidly develop new cells and tissues. The energy for growth and development comes from the endosperm, or the cotyledon. As you will discover in Activity 7.3.2, Seed Structure and Enzymes, enzymes convert starch into glucose in order to fuel seedling growth until true leaves are developed, and the seedling can produce food on its own via photosynthesis. As a review, mark or highlight 3 key points in your notes that are important to remember from this presentation. List 2 ideas or concepts that relate to previous knowledge. List any questions you have about this topic and discuss these with your instructor. Keep any notes you have from this presentation organized and available for use throughout this course. Through Activity 7.3.1, Baby Pictures, you will identify the structures of seeds and seedlings at various stages of germination. Comparing these structures will help you to identify the seeds and seedlings as either monocotyledonous or dicotyledonous. Your teacher will provide seeds and growth chambers for you to observe the various phases of germination. You will record your observations in a photo album over the next few days.